So welcome to this mini clip on phylodynamics. I'm Raphael. This is part of the tree thinking section of the crash course in evolutionary biology. So what is phylodynamics? Phylodynamics refers to the study of diversification rates, of rates of speciation and extinction when we're talking about species at the macro evolutionary level. But basically the study of the characteristics and the patterns that you see in phylogenetic trees. So in phylogenetics, we want to reconstruct trees, but once we have reconstructed trees, we may realize that those trees have certain features. For example, top right, you can see a plot that shows that uh, a very few genera of leaf beetles contain a massive amount of species, and then you have lots of different genera that contain almost no species. So there clearly are groups of organisms on phylogenetic trees that are more species rich than others, for example. What can explain this? That's what phylodynamics is about. So this term phylodynamics is not so used actually in the evolutionary biology literature. It's more used in epidemiology, where the phylogenies that are being studied are more phylogenies of viruses, for example, or strains of pathogens. But that's basically the same thing, like figuring out what are the causes of the different patterns we see in phylogenetic trees. How does diversification unfold, basically? So the first step to ask these kind of questions is, well, has been to come up with ways to first estimate the diversification rates of species, because this is really what we're trying to explain, differences in those diversification rates, for example. And so diversification rates typically mean speciation and extinction. And so these lambda and mu parameters that I'm showing here. So what is typically done is to assume that if you have a given phylogeny, that's the one on the left, uh, you can assume that this phylogeny has been produced by a certain process, a stochastic process maybe, like, well, in this case called the birth-death process, uh, where we imagine that each speciation event essentially corresponds to a birth event, and each extinction event corresponds to a death event. So if you imagine that this process unfolds through time, so this is the cartoon of the tree to the right, that through time some speciation events occur, through time some extinction events occur. So these are all the orange ones, the branches that never make it to the present because they become extinct. If you imagine that such a stochastic process may have given rise to the phylogenetic tree that you are observing, that you have reconstructed with your own data, and if this stochastic process that you're assuming is relatively simple, and here is very simple, it's a birth-death process with only two events possible, birth and death, speciation, extinction. So it only has two parameters. Then you should hopefully, and we've seen that already in the previous clips and also in the phylogenetics part of the course, you should be able to come up with a likelihood function for this model. So this model is this stochastic process that you're assuming may have given rise to your data. So that's essentially a model, relatively simple model with two parameters. The likelihood, assuming that you can come up with a formula for the likelihood, you should be able to get it. This likelihood is the probability of observing your data, and in this case, it's the tree that you have reconstructed, given the model of diversification that you're assuming. In this case, is this process that's on the right. Of course, you would, because it's a stochastic model that you're assuming, is a stochastic process. You would basically be assuming that the tree that you're observing is a realization of such a process. Now, if you can come up with that, then you can use maximum likelihood algorithms to maximize the likelihood and figure out what are the combinations of parameters of this model that best explain the tree that you're observing. And one important insight of this is that even though you might be working with a molecular phylogeny, so of only extant species, and you have no idea about extinct species because you don't have fossils, for example, you might still be able to recover from the shape of the phylogeny using a model like this the rate of extinction. You can infer this rate. Of course, is it well, literature says it's not very precise, and it's subject to all sorts of caveats, but the general spirit behind this estimation is this thing. So if you uh, were to visualize your phylogeny in a slightly different way and plotting through time in millions of years the number of lineages that accumulate from the root to the tips of your phylogeny. So every time a new node appears, a new speciation event appears, you basically increment. Uh, you should have a number of species that increases through time, right? As lineages accumulate. Of course, phylogenetic diversification is usually assumed to be a geometric process, so it grows exponentially, of course, because the more branches you have, the more branches are there to make new branches. So if you show this on a log scale, you should see a steady increase, a linear increase in the number of species. Now, that is if there's only splitting events and no extinction events in your tree. Now, if there are extinction events in the model that generated the data, you should see an increase in number of lineages that grows beyond the exponential rate that goes beyond this like linear increase on the log scale. And that is called the pool of the present. This curve is being pulled up as it gets closer to the present, so towards the right of the time axis. And that is because closer to the present, you have more species that will go extinct eventually, but have not gone extinct yet, just because it's so close to the present. So in this number of lineages that are included in this orange curve, you have a number of species that will basically that are guaranteed to go extinct. They just haven't done so yet. And so the difference basically between this curve and the straight line is essentially, in a nutshell, a representation of how many species are expected to go extinct. So algorithms that sort of estimate these species extinction rates use sort of this principle to sort of estimate. They use the ways to assess the features of this curve to assess to infer a speciation extinction rates using the likelihood function. But that's of course only the beginning of the fun, because once you have a model that can estimate species and extinction rates, then you can really start asking different questions about all kinds of patterns that you may have noticed in your phylogeny. So one of the most common patterns in phylogenetics or in macroevolution in general is diversification slowdown. So if you plot these lineage through time, again, the same type of plot that I mentioned before, that's called the lineage through time plot. Uh, you see that instead of a pool of the present, on most real phylogenies, you see a slowdown in diversification towards the present. And so that has led some people to suggest that maybe as uh, clades diversify and more species accumulate, then maybe they fill up all the ecological niches that are available to them, such that there's no more ecological space free to diversify more. And that process is called diversity dependent diversification. The idea that as all the niches fill up, there's less opportunity for new speciation events, and that's why on lots of taxa you see a slowdown in the accumulation of lineages through time. Well, then to detect a pattern like this, uh, or whether there is a pattern like this, some people have uh, expanded on the types of models that I showed you before to sort of be able again to use likelihood functions to fit those models to data and to tell whether or not there's a signal in a given tree that there is such a slowdown and that there is such a pattern of diversity dependence. And another very common pattern in phylogenetics and macroevolution is phylogenetic imbalance. Like we said before, some clades are much species rich than others, which is basically what phylogenetic imbalance is, the fact that uh, some clades have more branches than their sister clades. And imbalance taken to the extreme would basically look like this as a phylogenetic tree. Now, it could be that some clades have more species than others because they have a particular biological feature. For example, maybe carnivorous species that contain lions, for example, have a higher diversification rate than other species because of their diet being carnivorous. So you could be asking this kind of question, but if you were, it it would not be enough to just count the number of species that are carnivorous and the number that are not, or even divide this by the time that they had to diversify. You would need to use slightly more sophisticated methods that assume again some underlying models and that have likelihood functions for these models so that the model can be fitted to data. And these fitted models would basically assess or measure whether there is a correlation between the presence or absence of a certain trait, for example, diet or being carnivorous, and the rates of diversification in a certain portion of the tree. Now, the models that test for correlations like this between certain traits of the species and rates of diversification, so speciation and extinction rates, are called SSC methods or SSC models. That stands for state dependent speciation extinction.